Hello, welcome to my YouTube channel Forensic Forum. Our topic is types of antigen and antibody reactions. There are different kind of antigen and antibody reactions. For example, precipitation reaction, complement-fixation test, neutralization test, immunofluorescence test, agglutination, radioimmunoassay, and ELISA. On the basis of occurrence of the antigen and antibody reactions, it is basically two types, in vivo and in vitro. In vivo occurs in the human body or animal body, while in vitro are performed in the laboratory conditions. These are the examples of the reaction that are found in both the conditions. Let's discuss the precipitation reaction. In this reaction, soluble antigen reacts with antibody and forms insoluble precipitate. This type of reaction is called precipitation reaction. For this type of reaction, electrolyte, optimal temperature and pH is required. Agglutination reaction. In this, an insoluble antigen reacts with its antibody and insoluble precipitate is formed. This type of reaction is called agglutination reaction. For this reaction, temperature, pH and electrolyte is required. It is four types: slide agglutination test, tube agglutination test, antiglobulin Coombs test, and heterophile agglutination test. Complement-Fixation Test. For this type of reaction, indicator system consisting of sheep erythrocyte coated with amboceptor rabbit antibody to sheep erythrocyte is used. Complement can lyse these erythrocytes coated with antibodies. If complement is fixed and utilized in the antigen antibody reaction, there is no free complement to act on the indicator system. Hence, no lysis of erythrocytes occurs. In a complement fixation test, I am showing a two-sided image. One is the negative test and another is the positive test. Let's discuss the negative test. In this test, antigen, this is the antigen and complement is added to the microtiter plate, and we have added the sample without the antibody, and there is no complement fixation reaction occurs. In this, antigen and an antibody don't react with each other, so complement is free. After adding the indicator system, means antibody coated with the sheep RBC. This is an indicator system also called amboceptor. After adding these RBCs, our complement is free to lyse these RBCs. If there is a lysis of RBCs, means red coloration is observed in the microtiter plate. This type of results is showing a negative test. Against this, in the positive test, here this is an antigen and complement, and we have added the patient serum having the antibody, and antigen antibody makes a complex with the complement, and after adding the indicator system amboceptor, our RBCs don't lyse cause there is no free complement is there, and if there is no lysis of RBCs, this shows a positive test. When our body is getting infected by bacteria or viruses, these bacteria and viruses are able to produce exotoxins, and in result of that, our body is able to produce the antibody in the form of antitoxins, and these antitoxins are able to neutralize these exotoxins. This type of reaction is called neutralization reaction. In immunofluorescence reaction, a fluorescent dye conjugated to the antibody is used as a labeled antibodies, and these labeled antibodies are used to detect the antigens from the human body. Basically, two types of fluorescent dyes are used: fluorescein isothiocyanate, which exhibit blue color, lysamine rhodamine, which exhibits orange-red fluorescence color. Immunofluorescence is two types, direct immunofluorescence and indirect immunofluorescence. Fluorescence is the property of certain dyes which absorbs rays from one particular wavelength, that is ultraviolet light, and emit rays with a different wavelength, that is visible light. Radioimmunoassay. It is one of the most sensitive technique for the detection of antigen and antibody. In this, competitive binding of radio-labeled antigen and unlabeled antigen to a high affinity antibody occurs. After binding the labeled antigen to antibody, the test sample of unlabeled antigen of the unknown concentration are added progressively in a large amount. Here, two kinds of antigen compete for available binding site on the antigen. As the concentration of unlabeled antigen increases, more labeled antigen will be displaced from the binding sites. The radioactivity in the precipitate is measured from the unbound radio-labeled antigen. Enzyme-linked immunosorbent assay (ELISA). It is similar in principle to the RIA but depends on an enzyme rather than a radioactive label antigen. An enzyme conjugated with an antibody reacts with a colorless substrate to generate a colored reaction product. Such substrate is called chromogenic substrate. For example, horseradish peroxidase (HRP). ELISA is four types: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Here, spherical structure represents the antigen and green colored Y-shaped structure is primary antibody. Yellow spherical ball is showing enzyme which able to convert the substrate into product, and blue colored Y-shaped structure is secondary antibody, and in the sandwich ELISA red colored Y-shaped structure is showing the captured antibody coated in the microtiter plate. And in the competitive ELISA, sample antigen competes with a reference antigen for the binding to a specific amount of labeled antibody. The reference antigen pre-coated on a multi-well plate and sample is pre-incubated with a labeled antibody and added to the wells. If more antigen are there in the sample, less reference antigen will be detected and weak the signals. Please like and subscribe my channel. Thanks for watching.